Hello students, today's lecture is for the program Bachelor of Science 3rd year, subject Botany, semester 5, course code BOD 101, course title Plant Tissue Culture. Title of the unit, Unit 4, Types of Cultures, module name, Protocol for Cell Suspension Culture. I am Ms. Masha Rodriguez, Assistant Professor in Botany from Carmel College of Arts, Science and Commerce for Women. In this module, you will learn the definition of cell suspension culture and you will learn the protocol for cell suspension culture. By the end of this module, you will be able to define cell suspension culture and you will be able to explain the protocol for cell suspension culture. Let us first learn what is a cell suspension culture. Cell suspension culture is a type of in vitro culturing technique in which single cells or small aggregates of cells are cultured in liquid medium. The cells are isolated either from callus tissue or any other part of the plant in order to set up the cell suspension culture. Now we will learn the protocol for cell suspension culture. First, pour autoclaved liquid medium in sterilized conical flask. Then, inoculate about 3 to 4 pieces of pre-established callus into this liquid medium. Then, seal the mouth of the flask. Place the flask on a rotary shaker at 80 to 120 RPM at 25 degrees. Next, filter the suspension after 7 days using a sterile sieve. After filtration, centrifuge the filtrate containing the free cells and cell aggregates. Next, resuspend the cell pellet in fresh liquid medium and dispense into several flasks. Transfer flask to shaker and allow the cells to grow. Next, subculture at regular intervals. Plate the cells for organogenesis or embryogenesis. Then regenerate plantlets. And finally, harden and transfer the regenerated plantlets to the soil. Here is a diagram illustrating the various steps that are involved in setting up cell suspension culture. So here we have the pre-established culture which is aseptically cut into 3 to 4 pieces. Then this pre-established culture, here we have the pre-established callus which is aseptically cut into 3 to 4 pieces and it is inoculated in the flask containing the liquid medium. This flask is then sealed and placed on a rotary shaker with 80 to 120 RPM at 25 degrees. After 7 days, this suspension is filtered through a sterile sieve and the filtrate that is collected is centrifuged. Then the pellet is resuspended into fresh medium and then you pour that into several conical flasks which are then placed on the rotary shaker again. Then plate the cell suspension onto a nutrient medium, and then we have the regenerated plantlet, which is then hardened and transferred to the soil. So students, in this module you have learned that cell suspension culture is mostly initiated from a pre-established callus tissue, which is inoculated in a agitated liquid medium where it breaks up into smaller cell aggregates. This is followed by filtration, centrifugation, resuspension, plating, and finally the regeneration of the plantlet. These are the references for this module. Thank you, students.